Greetings everyone. In today's lecture of the part of the course Plant Physiology and Metabology, here we will delve into the topic of membrane lipids. We will explore the diverse types of lipids that form the structural foundation of biological membranes and delve into their roles and significance within cellular structure. Let's embark on this journey to understand the intricate composition and functions of membrane lipids in living organisms. This is the introduction that fundamental structural element of biological membranes consists of a lipid bilayer featuring two layers of lipids that serve as a barrier against the passage of polar molecules and ions. These membrane lipids possess amphipathic properties, meaning they have both hydrophobic and hydrophilic regions within their structure. The hydrophobic interactions between lipid molecules facilitate their assembly into bilayers, while their hydrophilic regions interact with water molecules. There are five main types of membrane lipids, such as the glycerophospholipids, comprising two fatty acids attached to glycerols, then galactolipids and sulfolipids, which also have two fatty acids linked to glycerol but lack the phosphate group found in phospholipids. Archaeal tetraether lipids, featuring two long alkyl chains connected to glycerol at both ends through ether linkages. Sphingolipids, consisting of single fatty acids attached to sphingosine, a fatty amine, and sterols, characterized by a rigid structure of four fused hydrocarbon rings. The hydrophilic moieties of these amphipathic compounds can range from a single hydroxyl group at one end of the sterol ring system to more complex structures. In glycerophospholipids and certain sphingolipids, a polar head group is connected to the hydrophobic component via a phosphodiester linkage, classifying them as phospholipids. In contrast, other sphingolipids lack phosphate but feature a simple sugar or complex oligosaccharides at their polar ends, categorizing them as glycolipids. Within these categories of membrane lipids, there is considerable diversity arising from the various combinations of fatty acid tails and polar heads. Glycerolipid, that the primary structural lipids found in the membranes are polar glycerolipid as depicted in the figure. These molecules feature a hydrophobic section comprising 16 carbon or 18 carbon fatty acid chains, bonded to positions 1 and 2 of a glycerol backbone. Meanwhile, the polar head group is linked to position 3 of the glycerol molecule. So, polar glycerolipids are divided into two main categories. The first one is glyceroglycolipids, where the head group is comprised of sugars, and another one is glycerophospholipids, that is characterized by a head group containing phosphate. As you can see in the figure, there are 9 different polar glycerolipid classes in plants, each of which can be associated with many different fatty acid combinations. Some glyceroglycolipids such as monogalactosyldiacylglycerol, glucosylceramide, sulfolipid, and digalactosyldiacylglycerol. Moreover, some example of glycerophospholipids such as phosphatidylglycerol, phosphatidylcholine, phosphatidylphenylamine, phosphatidylinositol, phosphatidylserine, and diphosphatidylglycerol, also called cardiolipin. In photosynthetic tissues, chloroplast membranes constitute 70% of the membrane lipids and are primarily composed of glyceroglycolipids. Conversely, other membranes within the cell predominantly contain glycerophospholipids, as you can see in the figure. However, in non-photosynthetic tissues, glycerophospholipids are the predominant glycerolipids found in the membranes. This is about the glycerophospholipids. Glycerophospholipids, also called phosphoglycerides, are membrane lipids in which two fatty acids are attached in ester linkage to the first and second carbons of glycerol, and a highly polar or charged group is attached through a phosphodiester linkage to the third carbon. Moreover, these lipids are built on a glycerol backbone structure with carbon atoms 1 and 2 esterified with fatty acids, while carbon 3 is linked to a phosphate group through a phosphoester linkage. This molecule is referred to as phosphatidic acid. The presence of nonpolar fatty acids at one end of the molecule renders it hydrophobic, while a polar alcohol group linked via phosphodiester linkage at the other end makes it hydrophilic. Depending on the type of polar group attached, such as choline, ethanolamine, serine, inositol, or glycerol, these lipids are classified as phosphatidylcholine. In short, it is denoted as PC, phosphatidyl ethanolamine, in short PE, phosphatidyl serine, denoted as PS, phosphatidyl inositol, denoted as PI, or diphosphatidylglycerol glycerol respectively. Phospholipids are integral components of membranes found in bacteria, animals, and plants. As you can see in the figure, depending on the type of polar group attached, such as choline, ethanolamine, serine, inositol, or glycerol, these lipids are classified as phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine, phosphatidylinositol, or diphosphatidylglycerol. This table shows the lipid composition in the plasma membrane, endoplasmic reticulum, and chloroplast membrane. Just have a look that each and every lipid has a different concentration in the plasma membrane as well as endoplasmic reticulum, whereas in the chloroplast, such as phosphatidylcholine is maximum in the endoplasmic reticulum, phosphatidylethanolamine is more in the plasma membrane as compared to endoplasmic reticulum as well as that of the chloroplast. Then the phosphatidyl inositol is absent from the chloroplast. This is one of the characteristics. Then phosphatidyl glycerol have a maximum in the plasma membrane, followed by chloroplast and endoplasmic reticulum. Then we have a monogalactosidyl glycerol, which has a maximum in the case of chloroplast and very less amount in the plasma membrane as well as in the endoplasmic reticulum, and so on. As you can see from the table, that distribution of different types of lipids in plasma membrane, then the tonoplast, nuclear membrane, endoplasmic reticulum, plastid membrane, mitochondria membrane, Golgi bodies, and glyoxysome membrane. So, in the plasma membrane, it will be the phosphatidylcholine, phosphatidylethanolamine, phosphatidylinositol, sterol, and its derivative. Whereas in the tonoplast, it is a phosphatidylcholine, phosphatidylethanolamine, phosphatidylinositol, sphingolipids, sterol, and its derivative. Then we have endoplasmic reticulum, mitochondria, Golgi body, as well as that of peroxisome. Every of the part of the membrane, they have the different type of the lipid. Some glycerophospholipids have ether-linked fatty acids. So, certain animal tissues and unicellular organisms possess high concentration of ether lipid, where one of the two acyl chains is connected to glycerol via an ether linkage instead of an ester linkage. These ether link chains may either be saturated, as observed in alkyl ether lipids, or contains a double bond between carbon atoms 1 and 2, as observed in plasmalogens. Vertebrate heart tissue exhibits a distinctive abundance of ether lipids, with approximately half of its phospholipids being plasmalogens. Similarly, membranes in halophilic bacteria, ciliated protists, and specific invertebrates also display elevated level of ether lipids. The precise functional significance of ether lipids in these membranes remain uncertain. It is possible that their resistance to phospholipases, which typically cleave ester-linked fatty acids from membrane lipids, plays a significant role in certain biological functions. Moreover, platelet activating factor and ether lipids serve as a powerful molecular signals. It is produced by a leukocytes known as basophils and induces platelet aggregation while also triggering the release of serotonin, which acts as a vasoconstrictor from platelets. As you can see in the figure, either ether lipids, plasmalogens, have an ether-linked alkyl chain, where most glycerophospholipids have an ester-linked fatty acids. Moreover, platelet activating factor has a long ether-linked alkaline chain at carbon 1 of the glycerol, but carbon 2 is the ester linked to acetic acid, as you can see in figure. It makes the compound much more water soluble than most glycerophospholipids and plasmalogens. The head group alcohol is ethanolamine in plasmalogens and choline in platelet activating factor. The second group of membrane lipids are those that predominate in plant cells, the galactolipids, in which one or two galactose residues are connected by a glycosidic linkage to C3 of a 1,2-diacylglycerol. Galactolipids are predominantly found in the thylakoid membrane, which are internal membrane within chloroplast. They constitute a significant portion, ranging from 70 to 80% of the total membrane lipids in vascular plants, making them possibly the most abundant membrane lipids in the biosphere. Due to the often limited availability of phosphate, a crucial plant nutrient in soil, there may have been evolutionary pressure favoring plants that synthesized phosphate-free lipids, thus conserving phosphate for more essential functions. Additionally, plant membranes contain sulfolipids, where a sulfonated glucose residue is attached to diacylglycerol molecule via a glycosidic linkage. The sulfonate group in sulfolipids carries a negative charge similar to that of phosphate group found in phospholipids. As you can see in the figure, there are the structure of monogalactosyl diacylglycerol or MGDG and diacylglycerol or DGDG, which are found in galactolipids of chloroplast thylakoid membrane. There are certain bacteria, archaea, inhabiting extreme environments characterized by high temperature such as boiling water, low pH levels, and high ionic strength, possesses membrane lipids with distinctive properties. These lipids consist of long chain 32 carbons, branched hydrocarbons linked at each end to glycerol through ether bonds. Ether bonds are significantly more stable to hydrolysis at low pH and high temperatures compared to the ester bond found in bacterial and eukaryotic lipids. In their fully extended form, these archaeal lipids are twice the length of the phospholipids and sphingolipids, enabling them to span the entire width of the plasma membrane. Each end of the extended molecule features a polar head group comprising glycerol linked to either phosphate or sugar residues. These compounds are collectively known as glycerol dialkyl glycerol tetraethers, in short GDGTS, which reflects their unique structure. Notably, the glycerol moiety in archaeal lipids differs in stereochemistry from that in bacterial and eukaryotic lipids, with the central carbon adopting the R configuration in archaea as opposed to the S configuration in bacteria and eukaryotes. This figure illustrates an unusual membrane lipid found only in some of the archaea. In this diphenyl tetraether lipid, the yellow box shows the diphenyl moieties, which are long hydrocarbon composed of 8 5-carbon isoprene group condensed end to end. As you can see, the glycerol moieties in archaeal lipids have the R configuration, whereas those in bacteria and eukaryotes have the S configuration. Archaeal lipids also vary in the substitutes attached to the glycerol. In the depicted molecule, one glycerol is connected to the diacaride alpha glucopyranosyl beta galactofuranose, while the other glycerol is linked to glycerol phosphate head group. This is all about today's lecture. Thank you.